Hello, everybody. Welcome. If you feel like it, feel free to introduce yourself, maybe where you're from, or what are you excited about genes? Or you can do it in chat. I'm currently in the UK, and Irish is joining from Toronto.
Mhm.
Hello.
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 I'm definitely very excited about genetics because, it's kind of the future of healthcare, and genetics just is an important part of our healthcare. And I'm sure Arashi will give perspective from the other end, which is from plants and biotechnology. And, she also has some experience in chemistry, right?
Mhm. Yeah. If you have any questions like about chemistry or the other sort of stuff, I'm more than welcome to ask.
Can you tell me how your day-to-day lab looks like as a, I don't know, geneticist or bioinformatician?
Sure, it's actually sometimes I'm just coding like 7 hours straight and nothing works. Lots of trouble. Sometimes I'm in the lab, pipetting things, um, getting things to work. Hopefully they work. Um, yeah, it's very varied. Most of the time, life in lab is a lot of failures. That's just the nature of science. And yeah, every day I look forward to something to not to work, and sometimes it does work, and that's a nice day. Mhm.
Yeah, I guess science, there's a lot of things that we don't know in the sense that whatever you see on papers are often like cool results, but actually the work behind the scenes, it can be laborious. Like you have to do the
Yeah,
same thing again and again and again, like pipetting and stuff. But I guess the results that you see probably are quite exciting, isn't it?
yeah, they're definitely very exciting,
Yeah.
and yeah, keeps you going when things work. And sometimes when they don't work, it's very curious, even though you know you did your stuff right. So that sort of takes you on a cool tangent, like, oh, why is this happening? This shouldn't be happening.
And tell me a bit about how you troubleshoot things. Oh, wait, Sakina said, um, high school student, high school junior, because I explore the world of genetics and learn more about bioinformatics research. Yes, what we really try to do is make sure whatever we explain hopefully will be accessible to everyone. Genetics and a lot of these bioinformatics can be quite complex if you read papers, but hopefully by the end of the session, you become a bit more confident that you can indeed learn. And in fact, you will hear Ayoshi's experience of how, if I'm correct, you were from the wet lab background and then you entered bioinformatics. And I think the cool thing of bioinformatics is you can analyze a lot of open source data set as well. So you can always play around with a lot of data. And she will also give a lot of advice on how you can get started. And yeah, that will cover a lot. Welcome, Sienna, Samuel, Mubashir. Hopefully I pronounced the name correctly. BP. Hello. Yes. Welcome. We will get started. I'm Sapo. I'm a PhD student at University of Oxford. And hello, everybody. Welcome to our seminar. We'll get started. So today, we will dive into different areas of genetics and bioinformatics research, from plants to medical genetics. And so we will cover very broadly of what genetics is, but we'll also go in depth to just give you a sense of something you probably never heard before. And some of the research we describe are also related to our personal research interests. So what that means is if you have any questions, you can ask and we can give our personal insight as to what it's like to be, a typical scientist. And it's my pleasure to welcome our guest speaker for today, Arushi Vardhan. Would you like to wave and say hello?
So,
That's great. Arushi is a graduate student at the Department of Cells and Systems Biology, University of Toronto. She's currently researching epigenetic mechanisms mediating seed development, and you will hear some of her fascinating research today. Her previous research experiences span bioinformatics, chemistry, nanoscience, integrative molecular biogeochemistry to tackle a wide range of questions related to biotech, climate change, and antimicrobial resistance. Again, as I mentioned before, if you have any questions about research related to, any of these topics, I encourage you to ask during the end of the session. And you can ask me as well. Complementing her talk, I'm also excited to share a different side of genetics research. I would talk about how genes are the building block of health and disease and give an overview of how genetic research like GWAS can elucidate the mechanism of disease and also help in the personalized medicine, which is kind of shaping the future of healthcare. So I will let Arushi take it from here, and she will mainly lead the presentation today.
thank you, Sapo, for that kind introduction. So let's get started. Today's seminar would be a gateway into the universe of genes. So, a brief overview of the seminar before we get started. I will start with different research area in genetics, and then I'll move on to how a certain subset of genetics called genomics is used in plant biology, and this will be tied into what I do in the lab and my research. And then we'll go on to medical genetics, which would be suppose talk. And then I'll go on to explaining different tips and strategies that I've found helpful to learning bioinformatics and different research skills. And we'll conclude the seminar with a brief Q&A session from you guys. So let's move on to research areas in genetics. Before I talk about the different research areas of genetics, I want to bring attention to the word model organisms. This is a word I'll use a lot in the seminar. So for instance, I use the plant called Arabidopsis thaliana in my research, also commonly known as tail grass. The reason we use these model organisms is because they're easy to grow, or they're more affordable, or the biology is really well studied. So if I want to ask a new biological question, I already have some context to work with. Also, a lot of tools and toolkits are well optimized for such systems. Similarly, a lot of people in gene research or research in general work with mice, C. elegans, E. coli, yeast, and fruit flies for the similar reasons. And sometimes, as Supper will also go into medical genetics, some of these model organisms are really useful because a lot of their genome is shared with humans, making them a good model to study human diseases and so on. So now I'll go on to genomics, which is one subset of genetics research. So genomics briefly is the study of an organism's entire genetic code, also known as the genome. So I, as a human, have a bunch of genes in me that encode what I am, and you guys also have a bunch of genes or a genome within you that encodes what you are like. And this makes us very alike and sometimes quite different. As you can imagine, this is quite a lot of data to work with. This is large. So genomics has something called bioinformatics and computational biology that we use to study big data. Bioinformatics and computational biology, the definitions can overlap quite a lot, but in a nutshell, bioinformatics uses tools to study biological data, whereas computational biology uses mathematical models and statistics to predict and study biological data. And we use these tools to study the structure, the function, the evolution, the mapping, and the editing of genomes. So as a genomics research, a question I would be interested in asking is, which genes are active under what conditions, and how do the products carry out those biological functions? I want you to look at this plot I've have right here, the circular thing. And I think this beautifully illustrates why genomics is so powerful. So this is a circus plot, and it's showing the rice genome. And OS stands for Arisa sativa, which is a scientific name for the rice genome. And 7, 8, 9, it's cut off here, 10, 11, 12 are different chromosomes of the rice genome. And what this is helping us see is the different segmental duplications that have happened in the rice genome. So if you see this orange overlap here between 8 and 9 chromosome, this is showing a bit of a duplication event that has occurred. And that's the beauty of genomics. You can take this large data, condense it into the circular plot such as the self, and understand biological conclusions and ask cool questions moving forward. And that's one of the reasons I really love genomics. Now let's move on to molecular genetics. Molecular genetics is perhaps the sort of genetics we're a bit more acquainted with. This is the genetics in the molecular, the scientists with the pipettes, and sort of that sort of thing. So molecular genetics is now going from that broad scale data of genetics, and we're honing in onto a smaller subset. So it studies how individual genes are organized, regulated, inherited, and expressed at a molecular level. So a Molgen scientist would be interested in asking, how is a gene structured to turn on and off in different tissues or in different developmental stages? Notice how specific this gets. And molecular genetics uses techniques in molecular biology and genetics to answer such questions. I think there's a good point to also explain. These days in science and research labs, no one is exclusively just using one research field to study a research question. We use a bunch of different fields, and when you hear my part of my research, you'll probably see how. But it's still nevertheless very useful to know different subsets of genetics research. Now let's move on to epigenetics. So epi comes from Latin and Greek and literally means above, hence meaning above the genome. And what this means in biological terms is external changes to the DNA sequence without changing this DNA code itself. So an epigeneticist would be interested in asking a question like, how do epigenetic chemical players such as DNA methylation or histone modifications affect gene activity without affecting the DNA sequence? And I think this image here illustrates this point very beautifully. So this circular thing here is a nucleosome with DNA wrapped around it, and out are protruding histone tails. And as you can see, the hands are making tiny like modifications at the side. And notice this is not actually changing the DNA itself. It's a little mark. And this mark has incredible significance in biological activity later on. And that's one of the really incredible things about epigenetics, sorry. And now we're going more into the realm of engineering and constructing things, and we're going into biotechnology. So biotechnology uses living organisms or biological systems to develop new technologies using principles in computer science, engineering, and biology. Two main subsets in biotechnology are synthetic biology and genome editing. So in synthetic biology, we build new biological components or we redesign systems using genetic tools. Imagine like you break your Lego piece and you start from scratch, and those building blocks one by one help you build something new. And that sometimes helps us understand these biological systems better. Or sometimes you can make new biological systems that were never even present in nature itself. And that's really incredible. You can do really, really cool things and influence gene expression in an organism with the power of synthetic biology. Another one is genome editing. With genome editing, we can use gene editing technology such as CRISPR-Cas9 to alter the DNA sequence itself. So let's say I'm a synthetic biologist. A question I would be interested in asking would be, can we create new genetic circuits that can reprogram plants to be better adapted to their surroundings? And basically sort of change the way Mother Nature has made them. Very cool. Now we're moving into medical genetics, and this is what Suppo will go into in more detail later. But medical genetics is studied with the motivation to cure diseases and help human health. So it studies the genetic basis of medical conditions such as cystic fibrosis, muscular dystrophy, and certain types of cancer. So a medical geneticist would perhaps be interested in asking something like, what genetic mutations cause inherited diseases, and how can we make targeted therapies to treat these specific genetic mutations? And now finally to agriculture genetics. Similarly, agriculture genetics is studied with the motivation of helping crops in the face of climate change. And this includes plant microbial and animal genetics to improve agriculture or promote traits in crops that are desirable for agriculture. So for instance, our agriculture geneticists might be interested in asking, how can we improve crop traits such as drought resistant, pest tolerance, and yield? So now that I've covered a broad overview of different areas in genetics, I'll go into a more specific one, which is how genomics is used in plant biology. And this is tapping into the realm of my research and what I do in the lab. So I'm a first-year master's student at the Satiki Lab at the University of Toronto. And we use epigenomics, genetics, and genomics to study seed development. Quite a mouthful. So I'll go into why we use this mysterious tissue called the endosperm. So seeds supply 2/3 of the global population's nutrition, and this makes them essential for humanity's needs. Within the seed, and as you can see this endosperm looking thing here, this is a placental tissue called the endosperm. It mediates the transfer of maternal information to the embryo. It doesn't actually provide any genetic component to the next generation of the plant, but is an important player in mediating this information transfer. A cool thing about the endosperm is that it's in a 2-to-1 mother to father genome ratio. It's triploid. So to give context, humans like you and I have a 1-to-1 ratio. I got one part of my genetic material from my mother and the other one from my father. But in endosperm's case, two of its genetic materials come from the mother and one of them come from the father. And this results in some really cool epigenetics going on. So endosperm's role in controlling nutrient allocation and providing environmental information makes it a potential site for something called parental conflict. A manifestation of parental conflict is known as genomic imprinting. So what is imprinting and how does this work in the endosperm? Now remember, let's imagine we have two genomes, one from the mother and one from the father. that we've inherited, and they're exactly the same. But one is expressed differently depending on which parent it came from, hence the word parent of origin. And when I say a gene being expressed, what I mean is being turned on. So now let's get into endosperm biology and what imprinting really looks like. And to illustrate this example, I'll first start off with imprinting's, I guess, formal definition. So imprinting arises from epigenetic differences between the genetic material you get from your mom and your dad. So now let's look at this imprinted gene, non-imprinted gene over here. So the genetic contributions from the mother are being expressed, and the genetic contributions from the father in blue are also being expressed. And all of these are being expressed equally. There is no bias. It's just a normally expressed gene doing its own thing. But things get interesting when we look at imprinted genes, such as MEGs, maternally expressed gene. So you see the genetic contributions from the mother in red are being expressed with these arrows over here. But the one from the father is not being expressed, and there is a bias in expression depending on which parent it is. Similarly, with PEGs, with paternally expressed genes, there's a bias in expression. The contributions from the father are being expressed, but the maternal genetic contributions are not being expressed. And this bias results in imprinted genes, and hence imprinting is a parent of origin effect. So now we'll go into the biological basis of how imprinting works. And one of the ways imprinting happens is through something called DNA methylation. So we have our DNA over here, as you can see, and DNA has 4 types of nitrogenous bases in it: adenine, thymine, guanine, cytosine, ATCG. And how DNA methylation really works in a nutshell is that you have the cytosine base over here, which is this thing here. And in this cytosine base, you add a little mark, a methyl group, and this makes it a methyl cytosine. And in the end, this makes it a DNA methylation. And remember, this is a type of epigenetic modification. You're not changing cytosine at all. You're just adding a little mark on cytosine. And this has profound consequences, as you can see with imprinted genes, for instance. So now that I've explained what I do in the lab and like the different research we do, now I'll go into how we study these kind of things. So in my lab, we primarily use bioinformatics, and the sort of programming languages we use to study Bash, Python, and R. So I use bioinformatics, for instance, to study something called transcriptome data, which is RNA-seq data, methylome data, DNA methylation data. We also use programming to make new code or new pipelines, or we tweak existing pipelines for better suited for our data analysis purposes. In terms of wet lab, or wet lab is in the realm of pipettes and chemicals, I do a lot of genotyping. And what genotyping I use for in my purposes is to study mutants and identify them. So genotyping workflow would be, first I extract my DNA, then I amplify that DNA using polymerase chain reaction, and then I visualize that amplified DNA using gel electrophoresis. If you're interested in what this looks like, I also included an example of how I do this on the Clematis Discord. Other more plant-specific techniques that we do in the lab are seed dissections to isolate embryo and endosperm, pollination and emasculations for the next generation of the plant, and library constructions to create RNA-seq data and methylome data. So I've explained what I do, how I do it. Now I want to explain how this ties in with the bigger picture. So has anyone ever heard of something called plant memory? Feel free to type on the chat. Sapo, you'll have to help me because I can't see.
Yeah, simple yes or no would be nice. Oh, Sakina says yes.
Oh, cool.
Very cool. Yeah, I think most people said no. I'm glad
Okay.
one person said yes. That's very cool.
Oh, very nice. Okay. Well, if you haven't, get excited because this is what enticed me into plant biology. Plants apparently can adapt and adapt to an ever-changing environment and pass on what they have learned to their offspring. But this is pretty weird. Plants don't have neurons. They don't have brains like we
Sorry,
do.
just to interrupt, Sakina said when a plant remembers on how to adapt after a disaster.
Yeah, that could also come into something called priming. And yeah, you're right. That is in the kind of what plant memory is. So yeah, so plants can adapt to an ever-changing environment and pass on what they have learned to their offspring. And as I said, this is kind of weird because plants don't have brains, they don't have neurons. How are they making these memories? One example has been shown through what I've been talking about, and that is through epigenetic modifications, where plants can remember or forget things they have learned from the environment through epigenetic mechanisms. although this might be a rarer event. And that's what makes this line of research just incredibly cool. It's still being studied, hotly debated if plants can remember or do they forget. And now that I've mentioned plant memory, now taking all together what I've said, we as plant biologists in this field hope that we can study such epigenetic mechanisms in plants and or seeds, and we hope to use the foundational knowledge used in this basic research and apply it to engineering plants and seeds that are better suited for withstanding our ever-evolving climate. That's it from me. Now, Sapo, you can take it.
That's excellent. And actually, she mentioned some bioinformatics stuff, which she will explain some tips on how to get into bioinformatics towards the end after this. So now I'm going to talk about medical genetics. So as we know, gene is the building block of health and disease. And you know also that gene is called, the blueprint of life because they carry instructions for all our body processes. Genes dictate from how our cells function to how we respond to our environment. And each gene holds specific information, and together they form a complex system or network influencing our overall health. Genetic variations, even very tiny ones, can actually make a significant difference. And these variations may make us more vulnerable to certain conditions. It may impact how we respond to treatments. It may also explain why some people can be more resilient to certain diseases than the others. So by understanding genes in greater depth, we open up the possibility to not only predict and diagnose diseases, but to treat them in a way that's customized or personalized for each individual. So I want to give an example of genetics research in schizophrenia. And I know Arushi mentioned cystic fibrosis and cancer, but those are the things we have heard of. But I think genetic research in schizophrenia is very exciting because schizophrenia it's very complex and there's actually a lot of cutting edge research going on, and therefore I want to take you through this experience of what is really happening on the field. So schizophrenia is a complex mental health disorder characterized by certain mental health symptoms like delusion, hallucinations, anhedonia or lack of pleasure, and avolition or lack of motivation to perform goal-directed tasks. And like I said before, schizophrenia is a great example to demonstrate due to its complexity. And also, it's quite difficult to treat. But before we can even think about genetics aspect of schizophrenia, it's actually important to ask whether schizophrenia is heritable in the first place. Because if schizophrenia is only dependent on environmental factors and it doesn't involve genetics, then there's kind of no point studying the genetics, in that sense. So, for every disease out there, it's important to ask whether there's a genetic risk towards such disorders. So, how heritable is schizophrenia? Or in other words, how much does genetics contribute to schizophrenia compared to the environmental factors? And let's see how we can actually study the heritability of schizophrenia. So we can study the heritability of specific traits or conditions using twins. Twins is a natural genetics experiment that we have. But before we can dive into how twin studies are being conducted, let's cover some basics. So let's say if Alex and David are identical or monozygotic twins, what percentage of their genes do you think they share? So is it 0%, 25%, 50, 75%, or 100%? Anyone would like to try and take a guess? Anyone want to type in the chat? 100%. Yes, that's great. And that kind of makes sense, right? Because, they share the same genes. That's why they kind of look similar. But if Alex are non-identical and or you call it dizygotic twins, what percentage of their genes do they share? Any other guess? This is a bit more difficult. 99%, not quite right. 50% or 20, yes, that is actually correct. So non-identical twins are synonymous to being a siblings. So actually it's 50%. And the idea is a bit more complex, but it basically comes down to the fact that each parent can pass down one of the two chromosomes from each pair to the child. And because each child will randomly receive one of the two genes from each of the parent, any two siblings or two non-identical twins end up with about half of the same genetic material. That's kind of like why it's 50%. I was supposed to show the answers. Okay, but whenever we talk about twin studies, we must talk about concordance rate. And it's a bit of like a tongue twister, I would say, but I'll try and get you through it. So In twin studies, concordance rate is very important, and it's an important measure in determining the heritability of certain trait or disease or condition. And concordance rate is the likelihood that both individuals in a pair, such as twins, will share a certain trait or condition. And let's see an example of how this really works. So let's say we identify all twins in, let's say, a country like Denmark. Okay. So most of these twins probably would not have schizophrenia. But let's assume that we take an unbiased sample, like we are able to identify all of the twins in Denmark. Out of all of those twins, we will identify all pairs of twins where at least one twin has schizophrenia. So let's assume we have 100 of those pairs where at least one of the twins has schizophrenia. Then out of those 100 pairs, we would count how many of the identified twin pairs have both of the twins diagnosed with schizophrenia. So let's say we have 30 of the pairs of twins where both of the twins in the pair actually have schizophrenia. So we call them affected pair. Now, remember what concordance rate is? Concordance rate is likelihood that like both of the twins will share the same trait. So here, the concordance rate is basically 30 affected pairs divided by 100. So it's 0.3. Or in others' word, there's a 30% chance that both individuals in a pair, such as twins, will share a particular trait or conditions, and in this case, is schizophrenia. So keep that in mind, okay? But now how do we use this to determine heritability of something? Let's establish three important points for this. So the first, as we know, identical twins share 100% of the genetics materials, but non-identical twins only share 50% of the genes, more or less. But the idea is non-identical twins don't have the same genetic materials. But we know that it's likely that both of the twins will be raised in the environment, and that's just an assumption we can make, though like if you do actual research, there's some ways to correct for nuances in the environment. But let's just assume for now that all of the twins live in the same household and kind of share the same environment. So the idea is that if the identical twins show a much higher concordance rate than non-identical twins, then we can say that schizophrenia is highly genetics. But why? Let's think about it. When genetically identical twins have higher probability of sharing a disease compared to non-identical twins, what that means genetics must play some role into this, right? Because genetically identical twins have basically similar genetics. But I think if I explain this, the last sentence here, I think you might understand better of what it means. If identical and non-identical twins show kind of very similar concordance rate, then we see that environmental factors kind of play a greater role in schizophrenia. So imagine now that genetically identical twins have the same probability of sharing the disease as the non-identical twins who only shares 50% of the genes. This means that genes are not really playing a bias or it's not playing a role. Because despite the fact that both of identical and non-identical twins have like different like genetic makeup, like where identical twins shared like 100% of the genetic material, where non-identical twins share only 50%, but yet they still share the similar concordance rate. So what that means is genetics doesn't really matter. And because both of them are in the same environment, so we would assume that environmental factors play a greater role. So what do the data show? So actually I got this data set from a really recent study in Denmark, actually. And they found that the concordance rate for identical twins for schizophrenia is 33% as compared to 7% for non-identical twins. And the idea is since genetically identical twins have higher probability of sharing schizophrenia compared to non-identical twins, that means genetics must play some role in schizophrenia, and what that means, schizophrenia is highly heritable. And actually, schizophrenia, one of the mental health disorders where genetics actually play a lot of role, and that's why there's a lot of research trying to understand genetics. So exciting. But now that we have established that genetics are an important factor in schizophrenia, we would ideally want to know, right? Like which of these genes in our body actually increases the risk of getting schizophrenia. And that's important. We want to identify those genes. And there's a technique that does precisely what I just said. It's called GWAS, or genome-wide association study. And as the name suggests, that's what it actually do. GWAS looks at our entire genome to identify genetic differences linked to or associated with a disorder like schizophrenia. And actually, the most common type of genetic variation in humans is SNP or SNP. So we'll call it SNP. And SNP stands for single nucleotide polymorphism. And if you recall, Arishi has shown that DNA sequence which have four types of nucleotides or bases. So we have A, T, C, and G. So a SNP occurs when a single nucleotide like A, T, C, or G in a genome sequence is altered. So for example, let me give an example. I think that might be easier. So let's say one person has an A in a particular position in the DNA, and another person has a G in that very same position. So you see that's a SNP because that's a single change in that particular position. And in human beings, we have such kind of variations. And actually, many of these SNPs or variation can influence whether you are more risk to getting that disease or not. So actually, it's very cool that just a tiny change can actually make you more susceptible to schizophrenia or like other disorders like Alzheimer's and so on. So to conduct GWAS, there is a standardized procedure to do this, and this kind of similar to like bioinformatics, you can say. And we can categorize the procedures as follows. So first we recruit participants, obviously. We need to recruit human beings, and then we extract the genome from the participant biological sample. Once we do that, we do quality control or we pre-process the genomic data, then we analyze them. And then lastly, we do association testing, which is you try to find associations between a genetic variation and a disorder. And I will go over this in more detail. So let's say we recruit 30,000 people with schizophrenia or cases or patients. And we also recruit 30,000 people who are healthy or like controls. Then we obviously have to collect biological samples like blood. And then we do something called DNA extractions. And those are the wet lab technique that Arshia has kind of mentioned. It's quite similar in some sense. Um, then we do pre-processing of data or quality control. And kind of, I want to give you a taste of what really goes on when pre-processing this data or analyzing this data. So for instance, in the quality control, we actually try to remove something called monomorphic variants. And monomorphic variants are those positions in the genomes where there is no variation among the individuals or population. And if since monomorphic variants do not contribute to genetic variation within a population, they kind of provide no useful information for studies that are trying to identify associations with traits or disease. So just let me rephrase this. So remember that in GWAS, we try to associate genetic variation of each individual to a disease. So if there's no variation in that positional gene or monomorphic variant, which is basically no variation, then it's kind of not useful for us in GWAS. Then we also try to remove rare variants. So rare variants are often found in only a small percentage of the population, but because it's so small, this variation exists in a very small set of population. It's not large enough and it's not useful enough to be a reliable biological indicator of schizophrenia. And so we often try to remove rare variants. And remember, we're dealing with millions of variants. So we want to make sure that there's kind of less noise in the data and really identify the useful variants. And then there are kind of established codes out there, or like on GitHub or something, to check for genotyping errors. We have to ensure that the reported appearance of phenotypes like sex actually match the gene data or the genotype, like, the XX or XY chromosome, which is the genetic kind of makeup of male or female sex. You can also do something called imputation. So what happens when you genotype a sample population oftentimes is, we can directly measure only certain SNPs. So in this case, in this experiment, they were able to directly measure only SNP 2, 4, 5, and 6, but we weren't able to directly test the sequence at SNP 1 and SNP 3. But we want to kind of guess what nucleotide exists at SNP1 and SNP3. To infer this missing data, researchers would use the known genotypes of other SNPs in the sample to guess the unmeasured or untested SNP1 and SNP3. And there's like tons of software database and algorithm that help us infer this missing data. So we call this process imputation, which is trying to guess the missing data, I guess. And now, importantly, we have to test for association. So we can use, you might have heard something called linear regression, which is in a more simpler terms, like any kind of correlation you see, that's linear regression. So y equals to mx plus c. Obviously, it's more complex than that, but that's essentially what linear regression is. So linear regression, if there's a variable like height is a trait that's continuous, then we use linear regression, or we can use logistic regression, which is the same logic, but we use the logistic regression for variables that are categorical. So for instance, whether this person have schizophrenia or not, that's categorical variable, and you use logistic regressions. And you can use like a bunch of statistical software like R, SPSS, like stuff like that to do this. And then you can also remove confounding factors in our model. So like age, sex, ancestry, and other demographic factors. These are confounding factors that might not really relate to disease. So we try to kind of remove them. And there's a mathematical model that can do that. And it's like a click of a button, actually. You can also correct false positives because there's like millions of comparison or associations you're trying to make. And therefore, there must be a stringent criteria to ensure that the genetic variations that are associated with the disease is actually true and it's not like false positives. We also want to combine data from multiple cohorts together, and we call that meta-analysis. So that just makes things more reliable. And a quick tip actually is to kind of watch YouTube videos to do this. There are tons of resources out there, and Arshi will actually go on and explain this. But I would also suggest that you should read primers or reviews that introduces GWAS if you're interested, because GWAS is such a standard technique in clinical healthcare and actually I'm sure in plant sciences too. Like the resources I got from is actually from a Nature Review Methods primers. So it's a journal that literally bring in experts that describe like a new methodology in like really simple terms. Okay, the point of GWAS though is to create a polygenic risk scores. So the idea is that let's say I have a lot of genetic variants that are associated with risk to developing schizophrenia. We can kind of create that score. And if I do that, I might be at the end of the bell curve. So individual one probably have like really, really high risk of getting schizophrenia. The bell curve is basically saying people in the middle of the bell curve have average risk, but people at like the right end of the bell curve have high risk. So that's kind of what you can do. And the idea is you can actually use GWAS for treatment prediction or personalized medicine. And the idea is that, like unfortunately in schizophrenia, a lot of patients, like over one third of the patient actually fail multiple conventional treatments and they kind of get stuck in this healthcare systems for like 5 to 10 years. And that's quite sad to see. And we categorize them as something called treatment resistant schizophrenia. What that means is they're resistant or they are not able to get cured from a lot of conventional treatments. But the idea is, are there any genetic variants that are uniquely associated with treatment-resistant schizophrenia? And the idea is that if there are such differences, it might reflect the underlying genetics of treatment resistance or of like the reason why they do not respond to so many conventional treatments. But the second question that's kind of more clinical is that can we use these polygenic risk scores or genetic variants to really predict treatment resistance? Or in other words, can we use polygenic risk score to predict whether who's gonna fail a lot of conventional treatment? Because if we can do that, then we could have easily recommend them to a more like invasive or like brain stimulation or like other non-conventional treatment, rather than let them do like pharmacotherapy or psychotherapy and make them go through this healthcare system that will not help them. So if you can predict it, that's great. And the findings, actually very recent. They have found certain genetic variants that are differently associated with treatment-resistant schizophrenia, but actually the sample is actually quite small, so they're not able to say with confidence that we can really use this in clinical settings. But the idea is you hopefully combine more data using meta-analysis. You replicate with like independent samples. Schizophrenia itself is quite complex as a mental disorder. So there's like a lot of variations within schizophrenia, and there's a lot of research to be done, but this is something that people have been trying to do. And yeah, so that's it from me about schizophrenia. And now I will let Arashi talk about tips and strategies.
Well, thank you, Sapo. That was really interesting and insightful to learn about. So yeah, I'll be going into different tips and strategies on how I learned bioinformatics and how to get better with your research skills and reading the literature out there. Okay, so as Sapo mentioned earlier on, I was a wet lab scientist when I started my research journey, which meant I didn't know anything about coding. And the most coding I ever did was I took an introductory computer science course, which was pretty helpful. So in all, I had a long way to go, and these are the things that really helped me, and I hope you guys also find it useful. So YouTube has incredible resources, and I want to give a shout out to this YouTube channel called Bioinformatician. She has such great videos on RNA-seq pipeline construction or DEC2 and using different tools in R and things. And it really, really helped me get better acquainted with data analysis. For instance, like what I found really helpful is that these YouTubers have like small data sets, and you can learn to reprocess the data with them, and you kind of get a knack and intuition for how data analysis work. Another channel I want to recommend is Genomics. I follow them on Twitter X, and they have very useful tips on analyzing data or like how to be better at coding and things like that. And these have been really, really useful, and I still use them today and refer to their videos when I do my own research. Another point I want to draw attention to is in bioinformatics, I find more than knowing the answers to everything, it's more important to learn how to look for help. There's an art to searching the things you need, and you will get better at this over time. So actually, 99% of bioinformatics is actually getting stuck or actually some really niche problem you have, and you're like, oh, I don't know what to do. There's something's not clear. This is where searching is an essential skill to have. I used the BioStars forum. Chances are someone in 2006 had the same problem you did and they figured it out. Or chances are someone had a similar problem with something else and that had a light bulb effect on you, like, aha, I know what to do now. And that has happened a lot of times. So it's really, really helpful to go to these forums. Reddit's Similar Reasons has always been really useful for me. Also, I really recommend really reading the manuals from the tool developers. For instance, I use Starliner to map my short read data to a genome. And there are many like variations or things you can sort of tweak and play with in your Starliner. And reading these tools are very useful to make sure you're doing the proper thing that you intend to. Also, these tool developers have GitHub pages with an FAQ section. And a lot of people, chances have already asked that niche question that you have, and you can learn from them. Or if no one has asked that question, you can ask that question. Also, another really useful tip is try to find people who do sort of similar stuff to you and read the methods papers they publish or the methods section and try to understand why they did a certain analyses. I'm going to take a leaf out of my stats professor's book. And by that, I mean is that in data analysis and in stats, like it will spit anything out. You can do whatever you want. But the thing is to be a good informed bioinformatician. And that is where reading and understand why people do certain things, questioning why they're done, how can they be better is a very, very crucial skill to have. And it takes time. I'm also still learning. Also, a bit different, more on the coding side. It's really good to look at published scripts on GitHub and get an idea of what elegant coding looks like. So, when I started, and I'm still guilty of this, when my code can go up to like 1,000 lines, and it doesn't have to be that long. It can be a lot shorter. And this is what I mean by concise and elegant coding. And why this is important is that you want to avoid redundancy. You want to avoid the potential for human error when you're playing with code. And having an idea of what really good coders out there do makes you better at coding and makes you realize how you can make your system better polished, streamlined for others to use, and that sort of thing. And it's helped me a lot. And I'm still learning this. If you're new to coding, I also want to say if you're finding it really hard, like capital letters hard, it's very normal. I found it really hard. I took 2, 3 months to learn Bash itself and the world of bioinformatics. It's very normal. Stay resilient. It does get better over time. I also want to end by saying that having foundational knowledge in computer science courses, so I took a Python course, was very, very helpful because before starting bioinformatics, I already knew the computer part. Like I knew what a computer thought like, or I know like the logic behind it. So when I had to go to the biology and bioinformatics part, at least that was over. So I really recommend taking intro to computer science courses. And now on to research to get familiar with, resources to get familiar with research, sorry. So reading primary papers in science can be really hard. I still struggle with it. It's very niche, or there are more words I don't understand that I do understand. And if you're in the same boat, you're not alone. What I recommend a lot of times is if I have a paper I like, I look at the last author of the paper and I search that professor up and I see like they probably have a website and I try to see like what do they do? Like, what are they interested in studying? Why are they interested in studying? It gives me like, dips my toes into their field and makes me a bit more acquainted with what they do. Also, chances are these professors have blogs, perhaps, where they blog about their work, or a lot of academics use Twitter or X to share their work. So this is really helpful because any new research that comes out, they will have a Twitter announcement and they have like a small thread with like the main findings, why it's helpful. And this is really helpful to get acquainted with research and sort of get a general idea of what's going on. The next step I have is more specific to a journal called Science, but Science has something called Science Perspectives, which I always found really helpful. It's written for a lay audience and it captures how this research that these authors have presented fits into a bigger picture. And I found that really helpful because instead of reading a paper and thinking, well, okay, they found this, what's the point? The perspective really allows you to see why this finding was so influential to the field or like why it's such a big deal. And it's always nice to be a better informed reader through these things. And third point, and a very, very crucial point, if you're a beginner like I am, it's very, very good to read review papers such as, and I love Nature Reviews Genetics and annual reviews of plant biology because they're very, very comprehensive and they really go in depth with what the field is. So reviews essentially summarize the findings of the field, and how they do is that, okay, XY years ago we had XYZ questions, we have found XYZ things, and now we have ABC more questions to tackle. And it's really helpful to have this bird's eye view. You're like, okay, so we know this, we don't know this, and these are more questions we've had. It's very useful to be in that side instead of reading things and you're like, oh, I don't know where this fits in. So reviews are really, really good for that. And after you've done all this, of course, reading primary papers is one of the best things to do. These are papers that have an experimental design and methods and results. And these are what most people think of when they think of research papers. They can be very challenging to understand. They have a lot of jargon, very normal if you're new to the field. I also still struggle with primary papers, but I hope this tips help you delve into primary research papers better by, you know, progressively, slowly going step by step. Yeah, and I want to finish with an important tip that I wish I knew earlier on when I started my bioinformatics journey, or actually even my research journey. When I was a newbie to research, I thought like tools and techniques in science were all the thing. And they are. I mean, tools and techniques are really cool. But more than tools and techniques, it's very, very important you have built a strong foundational knowledge in biology, in chemistry, in statistics, and math to do the and ask the right questions and conduct well-informed bioinformatics analysis. As I said previously, you can do whatever you want. You can click whatever buttons you want. It will give you something. But it's very, very important to be good at these basic biology and chemistry and computer science stats, whatever you want, to be able to navigate this avenue. So for instance, when I do a bioinformatics analysis, it's not A to B to C to D final output. It's not like that. It's A, I can do B, I can do C, or I can do Z. So it's very important that I am well informed and I take the right avenue forward because I could do whatever I want. And well, that's not doing good science, right? You want to ask the right questions and make sure your science is informed. Yeah, and thank you. That's it from me. And if anyone has questions, you're welcome to give them.
Feel free to ask questions. Feel free to shout in the mic or put in the chat. While you think of questions, I want to add a bit from, I guess, the medical genetics perspective. So like what Arshia described about bioinformatics, when you do bioinformatics, you're actually answering new research questions just by teasing out, different parts of data, and you can touch on it too after what I say. But I think in medical genetics and actually in a lot of fields, you often have a really standardized way. So like especially in clinical stuff, the questions are quite similar and you can see a pattern to it. Like, are there genetic variants for a certain disorder? But I think in medical stuff, it's really important to understand the statistical implications of what you do. So the techniques will be the same, but I think, for instance, the way you recruit schizophrenia patients, or even the way you recruit twins, or the way you model environmental factors, just these small changes that have been like debate for over 20 years as to how you can actually correct for environmental factors. So I actually suggest, especially for medical genetics, you should really understand biology is important, especially for clinical related research. Really important to know the statistics and the implication of data collection, or like how you recruit participants, or like when you conduct clinical trials, just the fact that you randomly assign from one person to a certain treatment versus another, and how you're like how the participants will not know which treatment you're going to, like placebo effect. These things have a lot of implications and real implications for medical healthcare. So I suggest you know statistics well for the medical stuff. And obviously, it's always good to know biology, but especially if you are looking into more bioinformatics, like Arushi's kind of research, then please know your biology, chemistry well. But regardless of the field, it's always good to have foundational knowledge. But I just want to emphasize that for medical related research, statistics is like it makes or breaks your research. You want to explain actually why you really need to know the foundational knowledge? Maybe give an example of how you can integrate or like centromeres or like all kinds of biological
Um,
questions.
I don't know if I can explain it really well, but I'll try. When I've been analyzing a lot of, let's say, RNA-seq data, and I see an effect being shown through a certain trend in the graph or something, it's very easy to be like, oh, this is probably happening. Yeah, like XYZ is more expressed. Yeah. But it's very important to make sure that what you're seeing is not like a compound in some ways, or like is actually what you're saying is what you're really seeing. And this is where something called sanity checks come into really hand, or um, basically kicking your data around to the point that it shows what it does. Like that's kind of how I think of it. So with my supervisor, whenever I show result, he's like, "Have you tried checking this? Have you tried if this is having an effect on this?" And somehow I have to translate that into bioinformatics and do analyses that test for that assumption, that assumption, that assumption, that assumption. And sometimes we don't see that data. Sometimes it's contamination, or sometimes it's some other biological phenomena that might be happening. I know I'm saying this in a very like abstract sort of way, but it really does happen. And how do we circumvent these? You knew your biology, you knew your chemistry to even ask the right questions before just taking it for face value. Like, oh yeah, it's kind of like when you compare two things, in a, or like when you have a large sample size, right, in stats, and you get a significant p-value, like you'd be like, yeah, it's significant, but is it really? Right? You need to make sure you do the right stats. It's sort of similar to that in bioinformatics as well.
So Sakina asked, may I know what specific softwares are used
Yeah.
in retrieving the data required for the research?
A lot of times what you can do is you can do DNA sequencing. You can use Illumina or something like that, and you send it to a facility, your DNA, and they like do everything for you and stuff like that. Or sometimes there's published data that people already have. Or for instance, like people already have published endosperm data already out there, and I can ask a bunch of new questions from there. So how I would do that is I would go into SRA archive, I'll go to the data set I'm interested in. There's certain toolkits that can help you like retrieve that data into your you could say your terminal, your bash terminal, and then you can import it essentially, like download it, and then you can start working with it.
So Sakina, if you want, so I know like you're asking probably you want to get started in genetics, right? What I actually can do, I think maybe just type in the Discord channel if you can, like in research ask or something, saying like
Yeah.
remind Arushi she's on Discord as well, like, oh, you want to start genetics and that you want to learn how to like get the data and play around. And maybe Arushi, when you have the time, just compile like a YouTube video where
Yes,
bioinformatician
yeah, yeah, yeah.
shows a video of that she plays around with a small data set. I think that's a good start. And
Yeah.
from there, I think you will get an idea of what she's doing, like SR. Like if you are new,
Yeah.
you won't
I know what I'm
understand that.
saying right now is like a bunch of random things, and it's a pretty terrible answer to
Yeah,
be honest.
but
But
please
yeah,
make
that's
sure
why
to
I watch these videos. Yeah, they're really
yes,
helpful.
please make sure to because there's
Yeah.
actually like more than one way to get the data, even for open source stuff. So just make sure you ask in Discord just to remind her, and she will make sure to do it. And feel free to tag her or me as well. So Samuel asked, considering all the work that go into the study of genetics, what's the probability of a disease-free world?
Ooh.
Well, I guess that question is for me. So I would say actually, although I argue that genetics are building block of health and disease, genetics actually does not answer even half of of what happens. Even like diseases like cancer, actually, you will realize like genetics actually interact with all sorts of other factors like environmental factors. So if you're thinking of cancer, then genetics probably play a huge role. But if you're thinking disease as a whole, then no, definitely not. It's because genetics is not the only factor, right? And actually in medicine, you really have to, so actually in precision psychiatry or precision oncology, you really have to, there's like an argument that you understand a person as a whole. And actually the way you even the way you think about cancer, the way you think about a certain disorder like blood pressure or stuff, not everything is genetics, right? A lot of them are contributed by environmental factors, or some of them are psychological. And they're just very complex interactions that I think research will be ongoing. All we can do, I think, is try our best to understand how a disease are formed and keep researching. And also genetics is something that's quite complex. It's something that you have to use a lot of inference to infer certain mechanisms. And a lot of discoveries, if I understand correctly, is just observation of certain patterns of data. And you're like, oh, actually, there's something called small RNA, or there's something, this mechanism that's probably discovered through other sets of experiment too. I do not think we will ever have a disease-free world just from genetics alone. That is like, I would say, just because of so much complex interactions. And actually, for me, I'm really interested in interdisciplinary research, like how, different disciplines kind of intersect and help us understand a disease better. For that, you have to kind of go beyond genetics. So if there's no further questions, we'll end the session here. But thank you everyone for coming. I hope you guys learned something from this. Thank you. Okay, bye-bye.
Thank you. Bye.
Clematis Research Empowerment Hub. Empowering research for everyone.